A very warm welcome to everyone present here. Thank you all for joining us. We at Natural Remedies are really proud that you have joined us today, and we'd like to tell you a lot about how we are moving our science forward. So we're really happy to have welcomed you to our second edition of our Natural is Future webinar series. In this webinar series, we will be exploring phytogenics. This is the way product development takes place, how quality is checked, as well as how it is reached out to the end customer. In this second edition, we will be exploring the quality aspect. I am Dr. Anant, a product manager from Natural Remedies. We will be talking a little bit more in detail with respect to the validation and standardization of various phytogenics, and in special reference to our special phytogenic, the NRSPP. About Natural Remedies, well, Natural Remedies is India's leading phytogenic solution provider. We have stretched our arms across various species like ruminants, poultry, swine, as well as aquaculture. And Natural Remedies, of course, our science is backed by almost the hard work of almost 40 different scientists from different sectors, be it phytochemistry or animal health. We have identified more than 220 phytocompounds. And these phytocompounds, of course, have been published in almost 120 scientific journals. And these, of course, the hard work that goes in with research and development, that has paid off with by helping us with almost 15 patents and almost 100 monographs in different pharmacopoeias like the British, American, and the Indian pharmacopoeia. But let me just take you back with what had happened last time. We took you through a very journey, basically. We took you through a journey where we talked about the evolution of phytogenics. We had talked about how it had started out from the ancient times, the Ayurvedic systems, coming back to the medieval times, where we introduced new concepts, and then moving on into validation as well as standardization aspects of it. So these are what we call the modern phytogenic additives. Why modern? Well, they have a bit of, in fact, a lot of science in them with respect to how they're standardized as well as validated. And our science is NRSPP. NRSPP is nothing, well, it is nothing but a very well-constructed crude botanical powder that is inspired by Ayurveda as well as modern science. NRSPP is structured, or it is made to go through a stringent quality control process, which involves stringent standardization as well as validation. This is basically in gist about NRSPP. But today, we will be taking you a bit forward into how standardization occurs. For those who are wondering what NRSPP is, well, it is Natural Remedies Standardized Botanical Powder. But before we actually introduce our main guest who's going to take you through, I had a question, and it's time to poll. I would request you to take about 10 seconds to go to the poll option on your screen to answer the question whether you have analyzed any product that you've got in your farm. This can be physical, chemical, microbiological, toxicological, any form. Have you had the interest or any interest to analyze the product? Wow, it seems to be a very more of a 60-40 ratio. Well, it's kind of important that, of course, standardization or analysis is very important. And to take you through to the next part as to how different kinds of analysis can be done, I would like to call upon, or would like to introduce to you first Mr. Prashant D'Souza. He comes into natural remedies with almost 30 years of experience in the field of phytomedicine for animal health. About 50 publications and as well as a book chapter. He is the GM of research and development in the animal health wing of natural remedies. He's also a visiting professor at the University of Bologna, Italy, in the Department of Veterinary Medical Sciences. And he's also the member of the American Association of Advancement of Science. Dr. Prashant D'Souza, I would like to call you on stage.
Hello, everyone. Thanks a lot for joining this program. First of all, I'd like to extend a warm welcome to you. I'd like to give you today a snapshot, an outline of how the quality control is done in our company with reference to our phytogenic products, a brief outline about it. So this is the agenda that we have for today. I'd be explaining to you the nature and types of phytogenics. What do we mean by standardization of phytogenic additives? Why standardization is required? Then I'll touch upon some of the challenges that we face in standardization. Then I will take you through some general techniques of standardization. Then the 4 core pillars of standardization. And then I'd also like to give you the outlook and the future of standardization, the newer techniques that are there in the market that we have also started working on it. So this is in brief that I will be presenting to you on this particular topic. So let me start with the nature of phytogenic additives. What are phytogenic additives? Phytogenic additives are essentially botanical preparations, herbal preparations, which are designed for improving productivity and health of animals. So classically, the phytogenic additives have been used as growth promoters, mainly to replace antibiotic growth promoters. Mostly they have been essential oils, oleoresins. One of the very common example is the oregano oil, which globally everybody is familiar. So phytogenic additives have been used in this field of animal health, in poultry or swine, even in ruminants, largely to augment performance in broilers, in layers, in ruminants, for improving the productivity. Well, the phytogenic additives can also be used to improve health, a particular target area, like let's say respiratory health, as a supportive therapy, along with let's say antibiotics. They can also be addressed to certain specific problems in farm animals, or let's say immunity, for example. And one of the areas which is especially popular in India is liver health. Here, it's very common to find the use of herbal supplements to augment liver health, to counteract the liver problems in farm animals. So the utilization of phytogenics is quite wide spectrum, both for enhancement of productivity or address specific issues of health in farm animals. So that is in brief what is phytogenic additives. Now I'll quickly take you through the different types of phytogenics that are there. So we have the botanical dry powders, which are basically dried plant material, ground into, the plant material is dried like twigs, leaves, whatever the case may be. It is dried into a fine powder, typically about 60 mesh powder. And when you are standardized with proper controls, then they become standardized botanical powders. That's what we do in our natural remedies. We do a lot of research in the choice of the material, and then we standardize by applying many cutting-edge techniques, and we standardize them. Then the botanical powders can be then called as standardized botanical powders. Then you have plant extracts, which are basically decoctions, in a way I can say, an extract of the plant material, which could be extracted with, for instance, with ethanol, alcohol, for example, or water, or in some cases it could be some solvents like acetone also could be used, depending upon the nature of the plant extracts that are required. So the plant extract would be a subsection of the botanical powder. You would the solvent would pick up only some selected compounds. Then you have essential oils and oleoresin. Essential oil is basically hydro distillation. You distill the oil from the plants. And terpenes. The oleoresins are another class which can be obtained by solvent extraction. Like for instance, you can use ethyl acetate and extract oleoresin. These oleoresins will contain a combination of essential oil and the plant resin. Then you could have pure phytocompounds. These are quite popular in the Europe, for instance. You have products which are made up of, let's say, eugenol, thymol, carbachol, cinnamaldehyde, pure phytocompounds. They may be synthetic or semi-synthetic, or it could be natural. It depends on the case. So you have a spectrum starting from botanical powders to pure phytochemicals. If you compare them in terms of the integrity, the botanical powders are very closest to the nature because we are not extracting them. You know, the chemical integrity is maintained. As you keep on purifying it, extracting it, you know, selectivity happens. You know, you may only be taking only a section of the chemistry. Now, you could also have a blend of these things. You could have a blend of, for example, botanical powders with essential oil or a plant extract with essential oil. That depends upon the strategy of the phytogenic company. So, the choice of these formats is based on the research strategy of that particular company. So, at Natural Remedies, largely we use standardized botanical powders where we have the natural chemistry of the plant retained, and normally the different compounds that are there in its holistic nature, they exhibit a phenomenon called polyvalency. Polyvalency means the multiple compounds that are there, they influence multiple targets, biological targets in the body, like receptors or transporter proteins or enzymes. So you could have a kind of wide spectrum phenomena that is happening. So you could, since we know that disease is multifactorial, it is one of the strategy could be to have a preparation, have a intervention which is multi-targeted. So since botanical dry powders, they contain a larger spectrum of compounds, you could expect polyvalency or multi-targeted effect. So that is our research strategy. So with this introduction, I'd like to now go to the concept of standardization of phytogenic additives. So what is standardization of phytogenic additives? So there's an official, there's a definition by a famous, phytochemist, Dr. K.K. Bhutani. I would like to present this. It says like this: the process of prescribing a set of standards or inherent characteristics, constant parameters, definitive, qualitative, and quantitative values that carry an assurance of quality, safety, and reproducibility. So the ultimate goal of standardization is to produce safe and effective products. The results have to be consistent. So after we, let's say, develop, in the process of development, we select raw materials and we come up with a finished product. So we apply quality standards. We apply quality standards based on our research, both qualitatively and quantitatively, so that our end product is assured for safety and efficacy. Qualitative parameters could be qualitative TLC, the way the product looks, the colors, the smell, organoleptic characteristics. Quantitative characteristics could be as simple as an extractive value to assay of active principles, assay of markers, things like that. It could also sometimes include biological assays. So by using this total package of qualitative and qualitative standards, you could ensure safety and efficacy. So in a nutshell, standardization is having those optimal limits done by research at the time of development. We set these limits, and then later we use this optimized limits, both qualitatively and quantitative limits, to monitor our product in the course of production. So why do you think standardization is required? One simple answer is, if you don't standardize the product properly, you will have inconsistent results. There are a lot of reports about inconsistency in phytogenics. I believe largely those inconsistencies are because of the lack of scientific standardization. And since plant material are very complex substances, they have heterogeneous chemistry. There are different types of compounds, and as a result, it requires real expertise and a very good scientific setup to standardize them. So what are the challenges? Why it is required? Why the standardization is required? So these are some of the reasons. The quality of phytogenic additives can vary due to mistaken identity. For example, many plants can look alike. They could be the same genus, the species could be different. So then there's a very famous case with Stephania tetrandra in Belgium. It was a part of a slimming pill. And during production, the raw material sourcing by that company, some mistake happened. They had substituted unintentionally to a similar looking plant to Stephania. And nearly 70 people in Belgium, they developed a condition called interstitial nephritis, a kidney problem, because the plant which got substituted by mistaken identity was having nephrotoxicity. So that is a kind of implication that can happen if you are not sure about the identity. Then time of collection is another challenge. So the raw materials, the plant raw materials can vary in terms of their quality, in terms of their active principle. For example, one very famous herb, licorice, as you know, the sweet tasting herb. In this particular plant, there's a compound called glycyrrhizin, which is responsible for the sweet taste. So if you collect in May, you get a particular level of glycyrrhizin. It varies completely different. It is completely different in November. So the time of collection of the herb, almost for every other herb, this is important. So the time of collection has an impact on the phytochemistry. Similarly, the place of collection. See, the soil factors, the environmental factors, all have an impact on the phytochemistry of the plant. So where you collect the plant makes a difference. For example, in one of our product, which is one of our top products, we know where to get that from. If we collect the material from North India, we get a very different chemistry. From South India, we get a very different chemistry. So we have optimized where to collect it. The place of collection has an impact on the quality. Similarly, the age of the plant. You take turmeric, for instance, the curcumin content varies based on the age. Similarly, the variety. You may have a plant material which is the same genus and species, but the variety can vary. The cultivars can vary. You take the example of geranium, which is popular for geranium oil. The different cultivars are there which give different types of oil and different phytochemical profile. So the variety also matters. Similarly, after you collect the material, how you process the material, how you grind them, how you size them down, how you process it. Let's say when you're extracting, which solvent to use, how you concentrate it, how you preserve it. All these things have an impact on the quality. Similarly, storage of the materials, both at the raw material level and the finished product level. For example, let's say essential oils. If you don't store them well, they get oxidized. They can also get evaporated to some level. You can have losses. So there are similarly even the materials, even the herbal powders, the botanical powders also have to be stored in a appropriate way. Otherwise, they can catch moisture and there can be degradation, there can be fungal growth. The storage becomes important. Similarly, how you analyze the material is also very important. For example, you could analyze polyphenols using spectrophotometry, or you can use a high-end HPLC method. So the results can vary. Of course, it depends upon how you are standardized, but the method of analysis has an impact. So what I would like to drive a point here is, it's not that simple, the answers are not straightforward. There are many variables that influence the quality of the herb. Starting from collection to the finished product, there are multiple variables. If you make a mix and a total number of combinations, it could be some 100 variables. So it requires a real scientific setup, a very good experience and expertise to handle plant materials. I would say the synthetic materials are relatively easy because you're dealing with one or two compounds. The plant materials are complex chemistry. It is estimated a typical herb can contain dry herb can contain as much as 25,000 compounds. So there are many variables like the way I described, which can get into the complexity. That is why it is very important to apply the principles of standardization. Only then you can expect the product to have a consistency in effect. Otherwise, the inconsistency in phytogenics is going to continue. So at Natural Remedies, we are committed to apply the best of the science available to have the best standardized product, and we are on a continuous research and collaboration with scientists across the globe to improve our tools of standardization. So quickly, what are the challenges in standardization? Yes, as I told you, the complex chemistry is a challenge. The inherent phytochemical variation due to various factors. Many a times active markers are not known. That itself is a challenge. So you don't know what to standardize. Compounds are not commercially available. Even if something is known, you don't get that easily. Varying methods of analysis between labs can also pose a method, because all the methods are not available in pharmacopoeia. You would have a method developed, but you may not be able to speak to another lab because they don't have that method, because there is nothing published sometimes. So inconsistency in biological results, obviously because of these variations, you could have it. Lack of sufficient knowledge on how the product works can also complicate, because if you know how a particular phytogenic additive is going to work, that will give you clues about the actives, and then probably you could work on the standardization. Most of the time, this information is not available. Difficulty in establishing a link between the phytochemistry and pharmacology is also a challenge, and that requires special expertise. So basically, these are the challenges, but one need not lose hope. Phytogenics or phytomedicine broadly is a multidisciplinary science. You need chemistry, phytochemistry, you need pharmacology, you need in vitro bioassays, you need biotechnology, you need molecular biology. Once you apply all these sciences and expertise, then you can have a very good grip on what to control, what to standardize. So it is certainly a complex science. Now, I'd like to take you through some general principles or general techniques with reference to standardization. So one of the principles is selection. So if you want to standardize, you be clear about what to select, for example, which variety, which place to cultivate, what plant to use. So the principle of selection guides you in that. You don't just use any tulsi or polybasil plant. You look for the variety. So the principle of selection guides you in that. Elimination. This principle emphasizes eliminate what you don't want. For example, let's say you have microbiological contamination. Then apply the principle of elimination, get rid of the microbes, or find out how to minimize it. Similarly, foreign organic matter could be one of the challenges. So if there is an adulterant, get rid of that. So principle of elimination focuses on eliminating what you don't want. At the later part of the presentation, maybe you'll be able to connect with this. Similarly, control. So principle of control talks about applying control at the time of collection, cultivation, collection of the material, then also at the time of processing the material, and then the quality control. So by applying controls, scientific controls, you can ensure standardization. Many times the nature doesn't produce, no two apples are similar, right? So the nature doesn't produce like a robot. Everything is varying. So the principle of readjustment talks about, blending two materials, two raw materials. For example, let's say we are talking about a particular raw material. The target assay we need is 3% of actives. Now the batches, the material that we have bought from the market is 2.5%, but you have a 3.5% material. You could blend the two and get your target assay. So readjustment talks about combining the different batches of the same material to get the target assay. So by applying the principle of selection, elimination, control, and readjustment, these are broad principles which can be used to apply control in terms of standardization. Then I'd like to now touch upon 4 pillars of standardization, or 4 core pillars of standardization on which the standardization rests. So the first pillar is the identity. So what is identity? Identity is the genuineness of the plant material. As I told you, plant materials can look alike, and if you don't make a proper assessment in terms of identity, you could have a problem. So how do you ensure identity? We have many techniques, 3 levels of techniques. Of course, there are a lot of details are there, but I'll touch upon the 3 important ones. One is the macroscopic characterization. So every plant material has a particular form and shape. So macroscopic characterization talks about shape, size, color, surface texture, including the smell, the overall how the plant material looks. This can be at the fresh material level or also at the dry material level, because when our people are growing it in the farms, they need to have a reference. So we train them with this reference and provide the reference material, and we train them. If we are collecting our material from the herbal markets, the raw material markets, then also the reference is provided, and the people, the collectors, the procurement people are trained as to what they should look for with reference to the target material. So they are provided training on the macroscopic characteristics, because those are the people who will decide what to buy. So they are given training on how this particular plant material, both at the fresh stage as well as in the dry state, it could look. For example, here you can see amla or Emblica officinalis. Macroscopically, these berries, how they look, and when they are dried, how they look. Similarly, you can look at this plant, Andrographis paniculata, the fresh plant, how it looks, as well as in a dry state. So we use herbariums, we train people, and they are clear about what they should pick from the market. So this is the principle of identity. Then we also use at the lab level macroscopic characterization. So basically it is histology, plant histology. We take plant material, we section it, stain with hematoxylin and leucine, and then we can look for plant characteristics. So each plant gives you a signature, which gives a histological picture which will differentiate from other. So this is another tool that we use. Even for a dry material, you have powder microscopy. You can use powder microscopy, you can use specialized stains, and even in a dry state, you can distinguish between plant material. Then finally, the most important tool that we use in identity is the chromatographic characterization. So quickly, what is chromatography? Basically, as I told you, the plant metal contains different phytocompounds. You separate them on a medium, like a silica gel plate. So there you get a profile like this, the one which you see. The different compounds they separate, which you can visualize by adding a stain, by adding a chemical reagent, or by exposing to UV light. You get these bands which are characteristic for a particular plant material. Similarly, you can develop that using an HPLC. You can separate these compounds in HPLC. You would get a chromatographic profile. So this chromatographic profile is basically a fingerprint of the plant material, because there are different compounds and they would separate at different times based on their density, or what we call as the dielectric constant. They would separate and they would give a fingerprint to the plant material. No two plant materials will have the same fingerprint. So what you can see here, for example, you can see here, there is this green, you can see a particular product, PHYTOC. You can see the different bands. So this is very characteristic signature of PHYTOC. Similarly, some of our other products like Corin Plus would not have this. So we know by using this characteristic signatures or PLC fingerprint, we can distinguish products. So this is a very useful tool. Then consistency. This is another very important pillar of standardization. How do we apply, how do we ensure consistency? So we start right from the controls during cultivation and collection. So when we do our cultivation, we look at the variety first of all. What variety we have to choose, and which variety choose will be dependent on the R&D research. The R&D would have worked on different varieties, would have narrowed down to a particular variety, and that variety will guide the people during cultivation. Then the method of cultivation. So the method of cultivation would talk about how you actually grow. So we have cultivation packages done based on agronomical research, and we know what are the conditions that are required to grow a particular plant. What should be the soil factors, what should be the pH, what should be the minimum fertilizer profile, things like that. So that is standardized. because you cannot, if you are varying the fertilizer, the chemistry of the plant also would vary. Then place and time of collection is also optimized. Where you want to grow the plant, that is also identified. Based on all this information will be first available based on our basic research, and then that would flow to the people on the ground. Place and time would be important. Then we also do the post-harvest technology. After you collect, how do you dry the material? How do you process the material also becomes important. Some materials require sun drying, some materials require shade drying, some materials require assisted drying within a short window of time. So all this gets optimized. We also do something like field monitoring of the material, because we may want to have the information about the quality of the material right there in the field. So for example, here you see a refractometer. You could assess the quality of, let's say, Emblica officinalis. You just squeeze the fruits, get a little bit of juice, apply on the refractometer, you'll come to know the total solids or the strength of the material, the what do you call, I can say the sap, what is the total solids present in the sap, you'll get an idea. Normally we get about, for Emblica officinalis, about 13 to 15 units. Suppose a particular lot of amla is say 8 units or 10 units, you know that there is something wrong. Right there in the field you'll come to know. So these tools, we have many such tools which are basically very handy tools which can be used on the ground. We are now even we are trying to use for pesticides. For example, the Japanese company Horiba has come up with many interesting tools to check the pesticides on the ground. So by applying these, we can ensure, see, once you collect the material with some pesticide, later it becomes a headache as to how to process it. So suppose if you were to collect the material, test it on the ground, you have a better control. So thereby you can assure consistency. So consistency at the field level, by having a control at the field level, at the cultivation level, at the collection level, you can assure, you can impact consistency. Then after the material reaches the manufacturing premises or production houses, we apply controls there. We work in the background of FAMIQS and GMP certified. We are FAMIQS and GMP certified. So we apply the principles of these good manufacturing practices. So there are checks and measures. There are critical control points, for example, CCPs as we call CCP, right? The critical control point in the process, where we do process monitoring. For example, we do moisture checking, we do sieve analysis to ensure the finished product consistency. So we have inline line clearance tests, in-process tests, and documentation and control as guided by the quality system like FAMIQS and GMP. So these controls in the manufacture will ensure that you're producing something consistently. Just imagine if these controls are not there, you would not have a control in what you produce. You don't know, you may not be operating at the right level, and there will be variations in batch to batch. So you control this by applying controls during manufacturing. So here you can see, we start from raw material, we clean sort, it goes through different mills, pulverizer, then it goes through magnetic separator, and then, some products can get sterilized and then ultimately they are packed. So at each of these stages, we identify critical control points and we monitor these critical control points in a stringent way. And it gets documented also, and it gets audited. And if there are any lapses, that gets surfaced. So like this, there is a scientific mechanism by which we have a control on our production process. Then comes, how do we ensure batch-to-batch consistency? According to EFSA, according to US FDA, according to WHO, the gold standard technique for ensuring the consistency of plant material at the quality control level is the chromatographic techniques, which could be the thin layer chromatography or you can use HPLC. So as I told you earlier, each of these techniques give you a profile. The compound sheet separated on a TLC plate or an HPLC column, and you get a profile. This profile is characteristic for a particular plant material or a finished product. So by checking this fingerprint with a reference material, you can be sure about the consistency of the product that you produce. You can see here that in different cases, the bands are all matching with each other. These are all different batches. Different lanes that you see, these are all from different batches. As you can see, they're all similar. That shows that there is consistency. Suppose there is some adulteration. Suppose there is, let's say there is some particular herb has not been added because of a mistake in production, then immediately you will find a big variation in this profile. Then you know the product is not consistent. Similarly, in the HPLC profile also you can make out. So you can look for adulterants or mistake in production or any lack of consistency in the overall chemical profile gets reflected in the chromatography. So this is a gold standard tool, although now many more sophisticated techniques are coming, but this is officially accepted technique for ensuring batch-to-batch consistency. That is chromatographic characterization. Then comes the assessment of efficacy, right? So, well, what is the common sense says? How do you assess efficacy? Means you need to do the animal trials, but you cannot be doing animal trials on a batch-to-batch basis, right? So it is, first of all, not ethical and not practical as well. So we apply, what we do is we use surrogate assays. In our basic research during product development, we identify what kind of fractions of the plant material, what are the actives, what are the phytocompounds that are linked to activity, and then we monitor these aspects during our quality control. For example, in our product PHYTO-C, we know one of the putative active compound is gallic acid. So we measure gallic acid both at the raw material, that is the Phyllanthus emblica, and at the PHYTO-C level, that the product Phyllanthus emblica goes into PHYTO-C. We monitor gallic acid both in the raw material and in the finished product. We know what levels have to go based on our basic research at the product development time. So we monitor throughout the raw material as well as the finished product. Similarly, arsolic acid and olinolic acid, for example, in Tulsi, these compounds are important in terms of their efficacy for PHYTO-C. So we monitor them in the raw material and in the finished product. So by using the surrogate assays, we could actually have a control on quality. It's not just active compounds. We could even use the total category compounds like total polyphenols, total flavonoids, total bitter compounds, total alkaloids. The different group category compounds also can be assayed. And of course, these should have been linked to activity at the basic research level. And during product development, we would have established, okay, for example, in PHYTO-C, minimum 10% polyphenols are required. We would have established that. Then we go about monitoring this when we do the assessment in the quality. Similarly, you could also use extractive values. Extractive value is like basically taking a decoction from a material. I'll give a simple example. Suppose you have tea leaves. Normally, let's say if you put for, let's say 10 grams of tea leaves, some 100 ml water, you get a particular strength of the decoction. Now, next time, if let's say the tea leaves were bad, the same 10 grams will not give you the same taste because the extractive value has varied, the profile has varied, right? So this is what happens. So extractive values are very broad indications of efficacy and consistency, because if the extractive value, typically what they use is water extractive value and alcohol extractive value. Of course, sometimes petroleum ether, chloroform also is used to cover the entire spectrum. The extractive values would certainly differ if the contents in the plants are varying, and if the extractive values are deviating, it would have an impact on the active compounds also, because active compound is a subset of these extractive values. So it's a very simple tool, but a very powerful one. So by using extractive values, successive extractive value means you extract and serially extract with different solvents, the same material. Then by using active compound markers and also group category, you could be reasonably sure in terms of the efficacy. Okay, now we have talked about we need to monitor some compounds to monitor the efficacy. How do we link the active compounds to efficacy? Because you may get this in literature. Good number of times you can get, because there's so much work that is happening on plant material, herbal material, medicinal plants. You may know, okay, what is important in anthrographis or licorice. You have that information, but sometimes you don't have it, especially if you have used a particular plant and you have done, for patenting purposes, very selectively, you have picked up certain material and done research and not well reported. Then you need to know the active. So we employ a technique called bioactivity directed fractionation, where we fractionate the plant with solvents of increasing polarity. So what happens is the plant material gets partitioned. The phytocompounds get partitioned based on their dielectric constant. They get partitioned. Some compounds will come in petroleum ether, the lipid soluble ones. The ones which are soluble, which are polar, they go in water. The in-between ones, they come in chloroform and methanol. So we first separate them. You take in this example, Tribulus terrestris, very well known in India for as an anthelmintic herb, to get rid of the worms. So here what we did is we used a worm called Series-Phytogenics elegans. This a tiny little worm can grow on Petri plates eating E. coli. So we use this bioassay, Series-Phytogenics elegans assay, and we fractionated the plant and tested these fractions on this worm and calculated the ED50 value, the dose at which 50% of the worms would get killed. We identified among the 4 fractions, petroleum ether, chloroform, methanol, and water, we found the activity was resting in the methanol fraction. We discarded the rest, then we focused on methanol fraction, 50% methanol. Then we further fractionated this and tested these 2 fractions, methanol fraction, insoluble one, and the soluble one. We found the activity was located in the insoluble one, and in the soluble one, it was not there. So we discarded that. We further fractionated the methanol insoluble one into ethyl acetate fraction and the water layer. We found the activity was located in the ethyl acetate layer, and from the in the water layer, we discarded the ethyl acetate layer. By further separating the water layer, we got 2 compounds called tribulosine and beta-cyclosterol diglucoside. So from a plant material, we could arrive at these 2 compounds, which was first time reported. We published this in a very reputed journal, Phytomedicine. So this was a collaboration between the phytochemistry team and the bioassay team. So it was a marriage between chemistry and biology which led to these compounds. So by doing this type of bioactivity guided fractionation, that is, the fractionation is guided by a biological tool. By doing this, you can link the phytocompounds to biological activity. Then that becomes a high value information, and that can be used to guide your quality control process. So this typically takes about 6 to 8 months. It's a lot of hard effort for 6 to 8 months, involving not less than 4 or 5 people. Well, now it's time to poll. You will find some questions popping on the right side of your screen. For the mobile users, it would be on the bottom of the screen. So I'd request you to take 10-15 seconds to answer this poll question, please.
So I'd like to just inform you that almost 97% of the attendees today have felt that standardization will ensure efficacy in products. How do you further see that, in the case of efficacious products, how can we further improve upon standardization aspect of it?
Well, I think this aspect, one has to be clear about what controls you have at the various stages and how you optimize the actives, knowledge of actives, and both qualitatively and quantitatively, you should be having a good control about the actives that are present, and then that would help you to have a control on the efficacy of the product.
So you believe more controls could define a more efficacious product?
Yeah, you need controls right from the collection. Of course, you need to, in your basic research, which you do during new product development, all this data will be available. You will have controls at the level of collection, cultivation, collection, processing, then quality control. At all levels, you should be having, and you should be having an idea about what is working in the product, its mode of action, what category of compounds are working. The chemistry also should be understood, so you can control the chemistry. So these are some of the controls that would be available. There's no single answer. It requires control at different points.
Okay, over to you, sir.
Thank you. Now we have talked about the 3 pillars of standardization. Now we'll touch upon the assessment of safety. See, plant materials, as you know, the raw materials for phytogenic is the nature, right? So you get this material from the nature. So in nature, the plant material can get contaminated by different things like foreign organic matter, heavy metals, pesticides, mycotoxins, radioactive compounds, or microbiological contamination and plasticizers. So, for example, many plants, they could be growing at near the rice field. So even though you may not use pesticide on medicinal plants because good number of them are weeds and they don't easily get infested with pests. But the water from the rice field can be contaminating and you could have pesticide levels going up. So similarly, the mycotoxins, if the moisture level is high, then you could have, if you have not dried the raw material, you could have the mycotoxin going up. So there is every chance that good number of contaminants can get into the plant material. So one needs to have a proper control in terms of these contaminants. So as I told you, you have a variety of contaminants. I will try to explain each one of them. I'll just take a few examples, maybe mention few others. So heavy metals, as you know, heavy metals are linked to a lot of health issues. You could have a variety of organ toxicity if you consume any product with heavy metals. So there are limits specified by USP, EFSA, WHO, with reference to the heavy metal contamination. So we use high-end instrumentation like ICP-MS to control the heavy metal load in our product. We check for lead, arsenic, cadmium, mercury, and sometimes many others. But basically, we check these four, and we ensure the heavy metal contamination in our product is well within the official limits. So this is very important. Otherwise, you would have a product, phytogenic product with more heavy metals. This would be eaten by a chicken, and the chicken tissues would have this heavy metal, and then ultimately the consumer will have the toxicity. So it is very important to control these things to produce a safe product. And believe me, analyzing these contaminants in plant metal also is an expertise. You require high quality skills in terms of good quality manpower to standardize, validate the methods. It's because these are all present in ppm or ppb levels. Similarly, mycotoxins. If you don't dry the plant material after post-harvest technology, if it is not proper, if you don't have the right water activity levels, AW values, or the moisture, if the moisture is more than 10%, normally the plant material will pick up the saprophytic fungi. The fungi would grow on the plant material, and you would have the mycotoxins like aflatoxin or the variety of mycotoxins. Close to 100 different toxins are there, mycotoxins. So they can grow. The fungi can grow, and your plant material can be laden with these toxins. So we need to ensure the product is safe. So we do mycotoxin analysis using LC-MS. We also use immunoassays like Aflacard, which is much quicker and quite a sensitive technique. But our gold standard is LC-MS methodology. For example, here you can see the peaks for aflatoxins, different types of aflatoxins. So we ensure that our raw material and finished product is free from mycotoxin by doing a detailed mycotoxin analysis. Similarly, pesticide residues. Plant material can pick up pesticides, from the soil, from the neighboring farm, and then the pesticides can be a real issue. There are very strict guidelines specified in the Europe and many parts of the world. Of course, even in India, we have certain limits specified. So we need to ensure that the pesticides in our raw material and finished product is well within the official limits. So we use techniques like GC-MS-MS and ensure the pesticide levels are well within the limits. So once again, this requires very good expertise to develop the method, to standardize and validate. It requires very good skills. So we have a very expert team. We have very good quality analytical scientists who are experienced in the process of sample preparation, method validation, and method deployment. So they are very good at it, and we check close to 70 odd pesticides. So I have not created a slide, but I would also like to touch upon the microbiological aspects. So you know that if the product is contaminated by pathogens, enteric pathogens like Salmonella, E. coli, Klebsiella, or Proteus, it's a challenge. So we check our raw material and finished product for microbial contamination. We check for total viable aerobic count. We check for total fungal count, yeast and mold. We check for total Enterobacteriaceae count. We check for selective pathogens like E. coli, Salmonella, Staph aureus, Klebsiella pneumoniae, then Clostridium. We check for these selected pathogens and ensure our product is microbiologically safe. Similarly, based on the requirement, we check for plasticizers. We look for sometimes radioactive contamination. We look for these different types of contaminants so that the finished product is free from these toxic residues. This is very important in terms of food safety. Otherwise, we would have a very both the animal, the farm animal, and the end consumer would be at risk. So at Natural Remedies, we have our quality policy on the safety aspect, where we are very particular in terms of our raw material and finished product being checked for its quality in terms of safety and efficacy. So this is a typical COA for our raw material, where you could see, for example, this is for a material called Tulsi Ocimum sanctum, the holy basil, where we check for some parameters like description, organoleptic characters, presence of foreign matter, identification using TLC, moisture level, ash content, which all indicates the contamination by salt and by silt, silica contamination. You'll come to know with this. Alcohol extractive value, water extractive value, then measurement of selective active compounds like arsolic acid, oleanilic acid. So this is a typical COA. Similarly, we have a COA for our finished product, where we check for organoleptic characters, extractive value, TLC fingerprinting, measurement of active compounds, and the absence of the heavy metals, and the microbiological compliance is checked. So now I'd like to take you to some of the recent trends in quality control. As you have seen, the plant materials are complex mixtures. They are, as I told you, thousands of compounds are there. And it is very simplistic to say only a couple of compounds are involved. Current research has shown, especially by the application of omic sciences, it is not the case. It's not just one or two set of compounds are involved. Definitely, it's a very reductionistic and a very simplistic approach. It's much more complex. Even the so-called compounds which are not called as active compounds could be influencing the biological effects by means of potentiation or synergy, or maybe just aiding the absorption of certain compounds. For example, it is known that certain polysaccharides, which may not be directly involved in the activity, but they enhance the absorption of some of the active compounds. Hence, there is a need for what is called the holistic fingerprinting or holistic analysis of the plant material, or a global fingerprinting. So the science of metabolomics, for instance, is catching up very fast, and we at Natural Remedies are working on this. So metabolomics approach gives you a kind of holistic approach. You don't just measure one or two compounds or a set of compounds. You measure the whole chemistry and then try to make a sense of it. So just as I will not go very deep into it, it requires probably an entire webinar, seminar for this. One more session may be required. So we start with the plant material. We subject that to techniques like LC-MS or proton NMR, and then the raw data is processed and then annotated in terms of the category of compounds. You can do both weighted or unweighted analysis, where you can categorize them in terms of similar group of compounds. Just qualitatively, you can separate combination of qualitative and quantitative characteristic and plot what is called a PCA plots, principal component analysis, which looks something like this. Each of these spots represents hundreds of compounds, qualitatively and quantitatively. This would give you a kind of global fingerprint or a signature for each raw material and finished product. This is the latest now in the area of phytogenics and phytomedicine, where you can apply a very high level of quality control to ensure consistency, because you no longer look at just few set of compounds, because after all, we know plant products are complex chemistry, and how they act as a whole is important. So the tools of metabolomics helps you in achieving that. So here you can see that currently we would be take an example of Choline Plus. We have been applying LC, HPLC, HPTLC, LCMS, maybe looking for selected markers and applying quality control. Very soon you will find Natural Remedies applying GCMS, MS, LCMS, MS, NMR techniques, applying metabolomics, and coming up with PCA plots or what's called a schemometrics for quality control. This is the future. Okay, it's time to have you engage in one more poll question. So you will once again find your questions on the right-hand side of the screen, and the computer, those who use a laptop or a computer will get it on the right side of the screen, and the mobile users will get it on the bottom of the screen.
So we've had almost 95 to 96% of them say ensure food safety. Very great, actually.
Okay, that's very interesting. Now, I think we will move, we are at the end of the session. I would like to give a quick summary of the presentation. So we know that standardization is critical factor to consider. You have seen the complexity of the plant material and the many aspects that go into it. So many variables, starting from cultivation to finished product. We will have one more session probably on new product development. Then you'll even understand the complexity. So standardization is a very critical aspect to ensure consistency. That's number one. Take home message. NR-SBP undergoes stringent quality control processes to minimize variability and ensure safety. We at Natural Remedies are really equipped with the state-of-the-art techniques. We have very good scientists who are experienced in these domains, and we apply these techniques to ensure our products are safe and effective. So we understand these variations and apply these controls. So the future is exciting. The future, we would be using more of omic sciences and coming up with new tools for quality control. Thank you.
Thank you, Prashant sir, for your wonderful delivery and talking about NR-SBP. For friends, if you had any other questions, please send in your questions to naturespulse@naturalremedy.com. And I repeat, it's naturespulse@naturalremedy.com. We have gotten a lot of questions. I must tell you, we've gotten a lot of questions from our people, and I'd like Prashant sir to answer some of them. Due to the time constraint, we may not be able to take in all the questions. So please forgive us. We will be getting back to you within 2 to 3 days. So Prashant sir, the first question I'd like to ask you is, given the current situation of COVID-19, etc., and the kind of things that are happening, are there any products or any ingredients or phytochemicals that could help in at least improving immunity or anything like that, or trying to counter COVID?
Oh, okay. You mean for the human subjects? Okay. Yeah. Well, it would be certainly erroneous to say if it can cure or prevent, COVID. What is available is for instance, there are a lot of immunomodulatory plants. You take, for example, polybasil or Andrographis paniculata. These plant products can augment the immunity of individuals. And there's already good amount of publications where the certain plant materials, like polybasil or Andrographis or licorice is used, and the research has shown there is a significant improvement in both adaptive immunity and innate immunity. For example, NK cell activity, which are the first line of defense against the viruses, the NK cell activity goes up. So this is already, quite popular in Thailand. Even in India, our government is started promoting, recommending certain herbal decoctions to improve immunity. So by employing these herbal solutions, one can definitely augment immunity, and there is good amount of scientific evidence for that. And in case somebody is interested, I can pass on those references.
Great. So yes, there are herbs that will augment immunity.
Yes.
Next, a question has come from Weng Yi. How much is what is the difference when you compare plant material from different sources, mostly geographic, and how do we solve this and achieve standardization?
I think I told you that. See, when we do research, we have a new product development process which can take up to 3 years. During this process, we check material from different areas. For example, let's say we are looking for a polyphenol type of material. We want a material which would. So we would have checked the material. Sorry, some disturbance. Yeah, we would have checked the material at different areas, and we would have assessed the chemistry and pharmacology. So we identify what is significant, which area, and then later we source only from that area. This is the control that we apply. So once you have, from your basic research data, you will know where to collect, what to collect, how to collect, when to collect. So this will become the profile or the research data which will go into our SOP, standard operating procedures, to guide your process of production. Hope I have answered.
Right.
Yeah.
Thank you. Okay, there's one more question from TCTO. So whenever cultivation is taking place, what level of fertilizer used or pesticide used, and how do we counter that?
Okay.
How do we?
Basically, I am into, of course, I look after the entire research, but the cultivation, of course, there are experts. I'm not right now familiar with the exact NPK combinations that are used. They use organic fertilizers. Fertilizers are used. But coming to pesticide, I have a fairly good idea. See, most of the medicinal plants are weeds, and these weeds are quite immune to the challenges of pests. Generally the cash crops are affected, but I'll tell you, surprisingly, a large number of medicinal plants are not affected by pests. For example, you take holy basil. Nobody adds in Indian houses on the holy basil pesticide. Do you add in your home? No, right? So because these are quite resistant. So luckily, a large majority of our plant resources that we use don't require pesticide in the first place. If at all that is required, then probably we also use neem oil and some, plant preparations we use. In case there is a need to use, then it is used in a very controlled manner, and we ensure that there are no residues.
Okay, great. So yeah, so we do have some standards for that.
Yeah.
So how do you? There's one more question. How do you exclude the effect from non-targeted active components? So how do we eliminate any kind of non-targeted active components or any interference in the phytocompounds?
Non-targeted active components. You mean active components hitting the non-targets, or what exactly? I'm not able to get this very well.
Okay.
Can you repeat the question?
How do we exclude the effect from non-targeted active components?
I guess the question is, if the plant metal has got some compounds and it would be influencing certain targets which is not intended for. Probably, I think that is so that's called as, in drug discovery, called off-target effects. So off-target effects would be possible. See, if there is some component in the plant metal which is certainly going to have an off-target effect, then we try to eliminate it by extraction or by eliminating the plant itself. We try to, so normally we use polyvalency 2,3 herbs, so we can, exclude it, or sometimes we can extract it and remove that component. But in the end, what matters is the end effect, phenotypic effect. Okay? If there are some off-target effects in the process, it doesn't really matter for us. What we ensure is the end phenotypic effects are maintained consistently. And if we recognize in our research, especially in vitro and mode of action studies, that there are certain off-target effects, it's okay as long as the end phenotypic effects are maintained. But at the research stage, if we discover some off-target effects, we try to minimize that by elimination, principle of elimination.
Right. One of the nature.
Yeah.
Okay. So we'll take in one, maybe one or two more questions, and
Sure.
there's one question about what could be some of the what are the benefits, of course, of using polyherbal formulations. And secondly, what could be certain some expected side effects that we can expect in cases of.
So you want to know the benefits of polyherbal formulations, right?
As well, yeah.
Okay. See, one very interesting thing, the plant world offers some unique advantages. For certain conditions, for example, you have a chronic disorder, or even in animals, for example, liver problems, the problems related to liver health. There are not very good options available in the chemical world. So the plant world offers. You take Silybum merianum, the milk thistle in the Europe or in India. You have so many herbs, like Andrographis paniculata. For certain specific problems, the plant world really offers some very interesting, safe and effective options which are not available. And something which you want to use on a long term. In such cases, you may not want to, like a supplement, you may not want to use a chemical product. The plant world offers some unique, safe and effective options. So in terms of immunity, liver health, and you could get very interesting options. Even, for example, gut health, in growth promotion for animals, you could have plant options, the phytogenic options, to safely augment the growth without having much problem in terms of the safety aspects. So another component why the plant materials or the phytogenics are interesting is their polyvalent effects. What is polyvalency? As I told you, plant materials are heterogeneous. They contain many types of compounds. They exhibit what is called as multi-targeted effects. They don't influence just one target. And most of the conditions, both performance issues and disease issues, have got a pathophysiology which is multifactorial. So you would need an approach which is multi-targeted. You will be surprised, the modern medicine, modern drug discovery, if you were to now research recent literature, check the recent literature, the multi-targeted approach is becoming popular even for modern research. So multi-targeted action is one of the things unique that you can expect in plant, in used medicinal plants. Certain conditions are best addressed using medicinal plants. Well, everything that is green is not safe. You need to have real research, good expertise, good toxicological assessment. Only then you can say that, and then good quality control procedures to look for unwanted materials. Only then you can ensure safety. Otherwise, you could have adverse reaction from plant material also. Anything that is green is not safe. The green is safe when you apply good quality science and good quality system. Only then it is safe.
Absolutely, sir. So standardization is very important to prevent any such kind of side effects.
Absolutely.
Finally, this is the last question, and I'm truly apologetic to the other audience members who have sent in a lot of questions, but we'll be taking one, and that's the last one that was sent. So during the process of standardization, do we expect certain nutrients in plants to be affected? Or do we see some kind of a change in their the composition of phytochemicals in the plant? And this can also relate to the different processes like drying as well as blending, etc. So do you see that?
First of all, nutrients, you use the word nutrients. See, in a plant material broadly, you will have what's called primary metabolites and secondary metabolites. The primary metabolites are your sugars, lipids. We are not interested in that. When we do phytogenics, we are interested in the secondary metabolites like polyphenols, saponins, alkaloids, terpenes. We are interested in that. So we monitor these, and that's where the standardization comes in. For example, for instance, if you dry the material at 60 to 80 degrees, let's say there is degradation of some actives, then we do much lower drying. We do shade drying. Right? So we will be monitoring the plant behavior of the plant material, both at the raw material and the finished product. We do stability studies, we do analysis during processing, and come to understand the settings, optimal settings, which will ensure consistency, which will not allow degradation. Plant material also can degrade. For example, I'll give you take Andrographis paniculata, andrographolite, degrades at a particular temperature. If you go above around 100 degrees, it will degrade. So you should not have any procedure which is around that temperature. So that's what I said. Standardization requires a lot of technical expertise, and by applying this, you know what are the right settings, what are the controls that are required that have to be implemented, and this data set will be used during your manufacturing process and your quality control process. So all this information you'll come to know during your product development, which, as I told you, takes typically about 3 years. During this, all these controls are understood and these are applied during production and quality control.
Right. Okay. Thanks a lot, Prashant sir, about enlightening us about how standardization is actually a very crucial aspect of phytogenics and what NRSPP is actually adding value to natural phytogenic products. So I would like to extend my gratitude. Thank you for taking your time, and thank you all for joining this webinar. Please look forward for more of such knowledge series webinars. Kindly follow us on different social media platforms, as well as www.naturalremedy.com. And also, just like to repeat, it's www.naturalremedy.com. Thanks a lot, and hope you have a good day.
Thank you.